Welcome to our virtual comparative genomics workshop. The Institute of Food and Agricultural Sciences, the University of Florida, and the National Science Foundation sponsor our research group to give 2 or 3 days workshops covering this material at the University of Florida and other institutions. IFAS, UF, and NSF have also sponsored the creation of this series of video lectures. This is the 6th in our series of videos, and it is entitled Creating Subsystems in Seed. The material covered in this lecture was prepared by Drs. Andrew Hanson, Valerie de Crécy-Lagarde, and Svetlana Gerdes. Drs. Hanson and de Crécy-Lagarde are both professors at the University of Florida. Dr. Gerdes is a member of the Fellowship for Interpretation of Genomes. She is a highly experienced genome annotator, well-versed in metabolism and familiar with the seed. The introduction, audiovisual material production, and editing of this series of lectures is done by Dr. Claudia Lerma Ortiz. We strongly recommend you to study our 4th and 5th lectures before proceeding to watch the material in this video. If you haven't registered in SEED yet, we remind you to open a personal account with the SEED RAS databases. This is free of charge, and it is in order to get a username and have access to all the neat tools offered by the SEED platform. To do this, please go to the home page of the SEED-based Rapid Genome Annotation Server RAST. Scroll to the very bottom of this page and click on the Register a New Account link. Follow the directions to apply for a new account. After applying, you need to wait for a confirmation email before using the newly created account. Since it might take up some time to obtain the requested account, we suggest that you do this as soon as possible. If you want to practice the exercises that we explain in this lecture right away, we recommend you to defer watching the video until you have completed the registration with SEED. If, on the other hand, you do not have a user's name but just want to get a general idea about the seed, or you have already completed your registration some time ago, please stay with us. Here is our lecture outline. We begin the 6th lecture by logging in the Subsystem editor. Then we visit the annotation overview page for different proteins, which we use as starting points to teach you how to locate specific protein encoding genes, and more importantly, how to annotate genes properly. The function of the entire SEED platform depends on precise and consistent annotations of all the functional roles of the genes in its database. Hence, it is imperative for all the users to follow strictly defined rules when doing these annotations. These rules are not difficult to follow, and we explain them thoroughly. Subsequently, we give you step-by-step instructions on how to build a new subsystem. We guide you in the registration of it, the choice of relevant functional roles, and the elaboration of a list of such roles. We teach you how to select and add genomes to the emerging subsystem, and finally, we show you how to generate a useful spreadsheet. Lastly, we teach you how to manage your subsystems, save them, find them, and even how to delete them if you no longer need them. If you need more information about the seed, please read the following paper by Rose Overbeek et al. 2005. The Subsystems Approach to Genome Annotation and its Use in the Project to Annotate 1000 Genomes. published in Nucleic Acids Research, which you can get in the link below, or go to the SEED platform main webpage with the link below. We will now proceed with our lecture. We recommend using Firefox as your web browser. To start the exercises, we need to go to our Comparative Genomics Workshop webpage, so click on the link below. We will also need you to open in a new tab the Subsystem editor, so click in the link shown below. Please type your username on the top right window and click the Login gray button by the window. Type folate biosynthesis on the window below the Subsystems name label and click Enter. You have now a small table with only 2 rows. The bottom row belongs to the subsystem done by Dr. De Crecy. Click on the link for folate biosynthesis. Click on the Spreadsheet tab. This will take some time to load. Once you get it, click on Ctrl F, and when the search little window appears, type Lactococcus lactis in it. The Finder will take you to the Lactococcus lactis rows. Click on PEG 1188-1. This will take you to the annotation overview for Fig 272623.1, which is the pyrophosphokinase GTP cyclohydrolase type 1 in Lactococcus lactis. Let's start now.
Okay, so everybody please find Lactococcus lactis foli, peg 1188. And when you found it, having got here, go down to the annotation overview, go down to the compare regions tool, and I want you, if you look at these genes, the peg we're on, which you can get by mousing over it, the peg we're on is 1188. I want you to move to 1177, and if you move to the extreme end here, that one is 1178. So I want you to move just one gene off the window. Well, there'd be a number of ways to do that. We could increase the region size, but the very easy way to do it is to just click this little arrow button, which, like all genome browsers, it'll click you one clunk along the genome and then it'll recenter you. So click that little arrow on one end of the thing, and this will recenter you, and you mouse over to find 1177, and 1177, we're just looking for this gene because it's an example of unannotated gene that I'm going to annotate with you watching me, because we can't all annotate it at once. So you move from the FOLE gene by clicking this to go one click or one slice of the genome to the left, and that's the gene we're interested in. So we only picked it because that one isn't really annotated. So now to recenter the display on that gene. So by clicking on it, and it's going to come up PEG 1177. Now notice that it has a general informative annotation, integral membrane protein. Let's just see how other proteins in that family are annotated. Increase your number of regions to 40 and hit draw. Now
How do I know that this is not annotated?
Sorry?
How do I know that it's not annotated from the picture?
Well, no, it is annotated, but it's annotated integral membrane protein, which is not at all a specific annotation. It's an honest annotation, but maybe we can do a little bit better if we, say, run the conserve domain tool, and that's what we're going to do. But just wait till you've got 40 of these, and just mouse over them all, and you can see, well, they're all called integrated membrane protein. So this is an honest, useful, but not specific annotation.
You can't add it like this in your subsystem. If you put integral membrane protein like this in your subsystem as a name, it would put thousands
it will
of
pull,
different.
good
so you
point.
Want something more specific.
You'll pull all kinds of unrelated proteins that descriptor would apply to in. So what we're going to do is practice putting a better annotation of it. Now, there's one last thing we're all going to do together, then I'm going to be like the police, hands on the top of the computers, because I don't want anybody clicking any buttons when I do it, because that would maybe cause a disaster. So after this point, as you might imagine, only one person can annotate the same gene at once. If 20 people try to change the name of a gene all at once, that won't have a good effect. So let's just do a conserve domain. Just click conserve domain, search for this protein, and it'll tell you that you can do a bit better than just integral membrane protein. It's been classified actually in several different protein family databases. We'll take the COG. It's been classified in the COG database as a particular family of integral membrane proteins, which is a more useful annotation, and as Valerie just pointed out, annotating it that plus integral membrane protein will allow you to specifically identify it and pull it into a subsystem. So that's what we're going to do, but this is where I want your stand out of the car, hands on the roof, please. Because otherwise, yes, I see you have experience of this, that it's otherwise we could cause a crash. So I'm going to go back and reannotate this as COG 4478. Notice what I'm doing. I'm putting this in a box called New Assignment, and then I'm going to add integral membrane protein afterwards because it's not wrong. And as long as we've got that in there, that's a nice specific annotation. And now I click Change. And within a second, it's now changed the annotation here to a COG 4478.
Click reload, with your browsers.
That's right. And if you all reload your browsers, you're going to see it's done that. Thank you. Now, know this. From my terminal here, I've changed the annotation in a computer in Argonne, and I've changed it for everybody. So this protein has now been renamed in the central server that serves everybody, and that means that if you had put it in your subsystem with the other name, sort of vague name though it was, it now would disappear from it because it doesn't have the same name, because subsystems work with names. So that's why renaming is a very delicate operation. And on the tutorial here, if I go back to that and just indicate this. Now again, please look at the screen here. Lots of genes have already been annotated and in some systems, a lot haven't. The one that we're looking at hasn't been annotated, it's not in a subsystem. We can annotate that, but you really shouldn't do it unless the gene has these characteristics, that it's not already annotated by somebody else and in a subsystem, and it's not really closely related to genes that are in a subsystem, such as, all right, the E. coli K12 gene has been annotated and the E. coli B gene hasn't. Well, that doesn't really mean you can annotate that differently from E. coli K12. That's a sort of common sense example. So use your judgment. Basically, you don't want to start annotating anything or re-annotating, I should say, that's already in somebody's subsystem, what you can do is just use their name. And we'll come tomorrow to ways of getting around names you don't like, or the fact that the gene may have been given different names by different people in different subsystems. There's a ways around this. The way around it is not to go in there and do a kind of clear cutting. I mean, you don't just go through and wipe it all out and put yours instead. Yeah, that's not how to do it. So we've now changed that. You've checked that it's been changed by going to the server. But suppose we now want to propagate that annotation, in other words, make all the genes that are closely similar to it have the same homogeneous annotation, which is obviously the aim of the exercise in seed. Well, there are lots of propagate annotations. We're going to see several of them as we work, but the one I want to start you with is just this annotate clusters tool at the top of the compare regions view. So again, don't touch this. Your hands are still on the roof of the car. And this is listing all the different genes that are in that view, starting with your integral membrane protein and going on to the other things that were in that view. You notice here it's called COG4478 integral membrane protein. Here it's just called integral membrane protein, but the homologies are very high. We can transfer the annotation from here to here by clicking that radio button on and checking these boxes. So always in transferring annotations with the different tools, it's always from the round radio button to the square box. You could even write that down in your notebook because that's a golden rule. There's no exceptions to that. That's the direction in which it goes. It goes from the round radio button to the square box that you check. And now we hit annotate, and in just a second or two it will reannotate the lot. They've now gone all the same colours. Theta was saying earlier on that colour is a way to tell you the annotation is homogeneous or not. And if we return to the feature itself, and we can close that box now. We go back to the feature, and actually you go back to the feature and refresh it. If I just refresh, you'll see that now that all the genes in that display. So mouse over all 5 of them, all the 5 red. You're having fun, good, good. All 5 of them have now acquired the same annotation. Now, this was your introduction to annotating yourself and then propagating the annotation to similar genes. There are several other ways to annotate and to propagate. We'll see those. But this is the basic operation, that you assign a name to a gene, one that you're sure of, and then you propagate it by homology and by the criterion as well of clustering if you're uncertain whether, is it in the same gene context as the gene that you're starting out with that you're sure is what it is. You use context and homology to propagate the annotation to other genes. And you're free to do that if your gene hasn't been already annotated heavily by somebody else, and if it's not in subsystems. And how do you know a gene isn't in a subsystem? Well, from this morning, you only know one place at least. Actually, you know a couple of places where information on subsystems is. Where's one of the places for the information on subsystems for a gene? Yeah.
String database? In string?
No, in the seed. Because we're talking, the seed, subsystems are specific to seed.
When you move the arrow over it, you see it in the list.
Yeah, that's exactly right. That's one of the most convenient ways. You just mouse over it, and it says it is in a subsystem. Well, it kind of is in one, but it's in one of these cluster-based things which are not real subsystems. Another way, simpler, is actually, if you're on the page, there's a whole section of this page which says whether or not it's part of a subsystem. And so sometimes, as we saw one this morning, it would be in a dozen subsystems. So these are ways to see whether a gene is in a subsystem. Well, now we've seen the basic operations of annotating genes. We're going to do, for the rest of the day, the other basic operation in seed, which is tougher. That's to build a subsystem. So we're each going to build our own small ones. Everybody can do this. I'd like everybody to do it. But when we come to adding genomes, we're going to be very prudent. Yeah. I would close this. I would leave that. And I want you to go to the subsystem editor, and when you go there, click Home. Go to the subsystem. Go to the subsystem editor and click Home. So then you should see all this stuff. So how are we doing? So you're still not on the page? We're all there. So you'll see a button at the top that says Create New Subsystem. Yeah, oh that's right. You still need to be logged in. If you're not logged in, this won't work. So you need to be logged in using your RAS username. Are you all logged in and on this page? Right, click the button that says Create New Subsystem. This opens a new window, and you need to give your subsystem a name, and I want you to call it your username, underline, and then folate. I want you to give them standard names so we can erase them afterwards, because these are practices and they'll just clutter the system if we leave them in there for years. So give them the standard name. Now, normally, if you were really writing, making a subsystem, you would have a description, so you would write something in here and you might write something in notes. Just put write test in both of those, just to give you the idea that you can, type in those boxes. And then, very important, all the way through the seed, there's going to be Save Changes buttons. And if you don't hit those, nothing will happen on the server. So to make this a real change, you have to click Save Changes. When you do, you'll get this little link at the top that will take you to your subsystem. And now we've opened in the subsystem to this subsystem information page, which has got test and test. If you wanted to, you can edit this. I'll put test 2 there, but if I did, save changes, otherwise it won't be kept. So now I'm going to save the changes, and to build, what goes into a subsystem are genes, functional roles, that is, and genomes. So we're going to add, first of all, one functional role and one genome. So to put a functional role in, you go to the functional role tab at the top, and you can put your functional role in here. Now, this is where this question of having exactly the right functional role, if you want to import something, if you get even one letter wrong, it will not work. So whenever you can, unless you're creating a totally new functional role, use copy-paste from somewhere else in the seed. So the first gene I want you to put in is GTP cyclohydrolase 1 FOL-E, from which you're going to get from Lactococcus lactis FOL-E by going to Lactococcus, and then capture GTP cyclohydrolase 1, copy it into the box. So whatever you do, don't just try and type that. Copy it from somewhere. From the subsystem, you could copy it by going to the folate subsystem, or you could copy it by going to any gene by that name within the subsystem. Yeah. When you're here, paste your GTP cyclohydrolase 1 in this box. Put an abbreviation in this box, and the abbreviation I suggest is FOL-E. All right, now when we've got there, you push Add Role, and then above all, Save Changes. So now we've got one role in our subsystem. Now we need to add a genome. To do that, leave this Functional Roles tab open and open Spreadsheet in a new tab. And the spreadsheet will actually have nothing whatever in it, because we haven't put a genome in there yet. So we're going to put one genome in there, and the genome we're going to put in would be E. coli. So go at the top here, we're in the tab that says Add Genomes, and all the genomes that are available in this seed are there alphabetically arranged in this pull-down window. You can scroll down, find E. coli K12 NG1655, and then there's a button under here which is fairly self-explanatory. Add selected genomes to the spreadsheet. So just click that. And when you've done that, scroll down and you will see a magnificent achievement. You've got the smallest possible spreadsheet with one genome and one function. This is of limited value for comparative purposes. But it is a signal achievement, and I congratulate you if you've got that far. Because now we're going to add some more genomes. So we're going to each add 10 genomes. What I want you to do that is to go back up to the Add Genomes and pick 10 genomes that you like. You can use the control button in this, okay? So if I like Acinetobacter baumannii, I highlight that, keep the control button pushed, and then go down and look for another genome I like, which might be, let's say, Bacillus anthracis. I leave that, and now pick 9 others from that list. And then add the selected genomes to the spreadsheet. And so when you push that button, you should see 10 genomes. So do you all have something that looks something like this? Different genomes, obviously. But if by any chance you don't see anything in these columns, the button that you want to push, we'll be doing this later more systematically, is refill all genomes. I mean, that's the safest one to push. By pushing that button, it tells the system to go out and look for all the genes in its collection that have the annotation for this gene that you put in your table. So it's saying go out and pull in everything to the spreadsheet that's got this annotation. And when you're just beginning with the first gene you put in a spreadsheet, it does that, usually does that by default. One person it didn't do by default.
I never understood why sometimes it does it, sometimes it doesn't.
But that's the fix, refill all genomes. So, okay, does everybody have this little tiny 1 gene, 10 organism subsystem? We're now going to add interesting genes to the subsystem. To do that, I want you to go to the E. coli, click on the E. coli gene, and
Which one?
which E. coli?
No, sorry. This already has 2.
The 1655 is the one I was meaning, but I don't think it matters.
Oh, it's fine.
And then ask for 50 homologs.
That's all just based on area, on alignment, on clusters, right? That's all cluster based. So if they're not close together, it wouldn't come up.
What wouldn't come up?
These maps. It's just
It's strictly looking. It's looking for the homolog of your gene. That's the gene we went in with. It's looking for the closest homologue, the next closest after that. And it's simply putting either side of that gene,
Adjacent.
8,000 base pairs.
If they have the same color, it means they have their homologue.
But if the homologue is like on another chromosome or something, it wouldn't go
No,
out.
it's just putting in the block of genes, the block of DNA, whether there's genes in it or not, either side of your gene of interest. Now, in most cases, if these are closely related, if this gene is closely related to that, there's a very strong chance that it's coming from a very similar genome, so all the genes in that block will be similar. But not always. Sometimes the gene is kind of on its own and you don't get this conserved region. So what I want you to do is scroll up and down a little bit here and notice in a few genomes, well, let's do something really simple. In E. coli, the gene next to GDP cyclohydrolase 1 is this putative inner membrane protein, which is not a very good annotation because it's too vague, as Valerie was saying. But the other side, there's a very specific one, S4-malglutathione hydrolase with an EC number. That's a very well-known protein. So why don't we put that in our subsystem and see how often that turns out to be clustered in any of the other genomes. That's the kind of hypothesis you could test. Is this gene clustered in other genomes besides E. coli? So to capture the name of that and put it in your subsystem, the easiest way is to click on it. It will take you there. Copy the annotation from
extra blank.
with no space, that's right, very careful not to do that, and put that then in your subsystem here and click on the gene, the S4-malglutathione hydrolase. Well, you can put any name you want. These are your subsystems. This is important. This is the part that has to be standard. This is the term that the system is using to identify the gene. In your own subsystem, you can call it what you want. This is not a term that's being used to search for anything. This is just a label you've chosen for it. So you don't have to slavishly copy what I put here. You can put anything you want. These are the ones that are operational. Yeah. So when you put that there, once again, add roll, save changes. When you've done that, go back to the subsystem and you now need to import that new gene and all the entries to the cells. So to do that, you just click Refill All Genomes and it will import that second gene and it will populate the cells that have that gene in them. So now you should see something like this, in which you have a column for this S4-malglutathione hydrolase, which is present in most, but not all of your genomes probably, because this is a common gene. And now, if you wanted to look at clustering, if we wanted to look at clustering, somebody tell me what we would do. Color it, yeah. Color by cluster. So we go up to the top of the spreadsheet, we go to the color spreadsheet, activate color by cluster, color spreadsheet. So now you also see something like this, but there won't be much clustering with these genes except in close relatives of E. coli. And there the clustering is probably just because of shared inheritance. They're close relatives, it's not functional. So you've all seen something like this. Right. Again, now is where I want you out of the car again. I don't want anybody doing this because I'm going to do another method of annotation to the one we've already done, to practice, to show you how to do another way. Now, I'm going to go here to the E. coli. In my spreadsheet, I'm going to E. coli fol E. And remember, we were looking at this and seeing that on one side there was this glutathione S hydrolase, which is a very well annotated and well known gene. On the other side is a thing called putative inner membrane protein. Well, this is another case where it's not really very helpful because that's far too vague, and as Valerie said before, if you tried to use that as an identifier, you'd pull in all kinds of unrelated things. So if we wanted to work with that, we'd have to give it a better name. If we mouse over it, we see it's not really in a subsystem. It sort of is, but not really. Certainly not in a string of them. So I'm going to click on that gene and ask for the conserved domain again, just like we did for the other membrane protein. And this is going to tell us it's, once again, does a very good job. It tells us it belongs to family COG2311, and that's a much more informative and precise objective annotation. So that's what we're going to call it. We're going to call it COG2311. And change it. And like before, if you want to go to that page and just open that page yourselves, don't change anything, just open it and look, and you'll see a display like this where it's, the top one is COG2311, all the others are something else. Now, if we want to propagate this another way, we can do it from feature evidence. Instead of doing it before, we did it down here, remember, from annotate clusters. You can also do it from feature evidence. It takes a second to open. So don't you do this. This is where, again, I'm doing it for you because we can't all do it at once. And you want the tabular form where, as Theta was explaining, the default here is the 50 closest genes, 50 closest proteins. The top one always has this sort of khaki color. You can assign from to everything in this table, and again you're seeing, as Theta told you, you've got e-value and percent identities which are really very high. So if we go right the way down to the bottom of this table, we see if the colors are different. Remember, colors are a way of telling you in this column whether you've got homogeneous annotations. These have got slightly different ones. COG2311, predicted membrane protein, but to all intents and purposes, that is not identical to the one we put in. Ours was called COG2311. It doesn't matter that that has that term in it. When C tries to pull it into a spreadsheet, it's not going to see that if we've defined it as COG2311, even though the word is there. It's not exactly the same descriptor. So all these are similar. Right down to the bottom, we've still got, you know, blast scores of over 160% identity. So we'd be perfectly justified in propagating that annotation the whole of the way down this table and actually beyond to further homologs if we wanted. Now, you could do this the laborious way, which would be to go and click every one of these boxes one by one. You'd propagate from this one, remember, from the round radio button to the square boxes. Or, if you want to do it fast, you can do check to the last box that's checked. So let's say that we're going to arbitrarily propagate this until we get to above 70% homology, let's say. So that's where 70% homology stops. So we're going to click that one. It's the last 70% homology. So I do check to last checked. And now it should, if it's working, have checked all the way down to the 70, which it did, and then not beyond. So that's a really fast way of doing it. We could have checked them all if we wanted. There's the all option up there. So having done that, then you see, I guess it's the assign function button, and then it will process, and it will change all of the names to this. Now you're beginning to see, once you have a well-defined protein family, or protein, you can propagate the annotation very fast, because we've just now done 50 genomes, and you could do hundreds of genomes that way for a well-defined protein that you were sure was real, really the same thing. Now scroll down, you can see all the things have the same white color until we get to below 70, and then we've left the old annotation in place. And you might be a little bit curious about these bright colors here, because Sveta was telling you this generally means, well, it means that there's only a partial match. And what we're seeing here for this one is only 79 gene residues out of our probe set of 300 and something matched, and there were only on all 79 in the targeted. So this is a truncated gene, possibly miscalled. It's a complete judgment call. I mean, there's a thing called the twilight zone, which is below 30%, is very doubtful if it is the same, although it still can be, but 30% is where you really start to worry that it might not be the same thing in terms of percent. But you can have things that are 95% identical that have a couple of catalytic residues different, and they don't have the same function either. So what's a much more faithful guide to whether you can propagate is actually context, is whether the gene is still hanging around with other genes that would suggest that it has the same function as they do. If you haven't got context and you've gone below, arbitrary numbers or 60% identity, you can't be sure. That's where the art or the judgment comes into this. I don't know, do either of you want to expand on that? Because this is the skill part.
I'm getting that to undergrads, that you can have 99% identity and it's still not the same function, and you can have 30% identity and it's the same function. It's all about other pieces of evidence. It can be clustering, it can be the presence of a motif, it can be a structure, it can be that the pathway is active in that organism. Annotation has to take all these in account. The blast score is just one piece of the puzzle.
One thing you can say with some certainty is that if you're dealing with genes in different organisms, a priori you can expect, and there's exceptions to this, but the more taxonomically distant the two organisms are, the less likely they are to be highly identical. So if you're going between plants and alpha proteobacteria, well, even the same isofunctional proteins may only be 40 or 50% identical. If you're talking about the same organism and you find two proteins in it that are 40% homologous to each other in the same organism, they probably are paralogs.
You want to do trees in that case.
You want to do trees, yes. See, so it's a context-specific question. If it's in the same organism, 40% homology is not impressive. If it's between widely distant organisms,
you're fine.
then it could well be very credible. In fact, it would be a good score,
it's a good score
if you had widely
it's really
different organisms.
So you have to start, you have to do it, play with the biology, find, that these are the same function, you look at their scores and you do that again and again, and after a while you kind of know.
But that's a really good question. It's the sort of thing that we need, we've been doing it for so long that it doesn't really come to the surface.
But this is something my undergrads, I try to get that to them and say, and at the end of my blast lecture they say, so what's the cutoff? And I'm like,
So that's why SID gave up any cutoffs altogether. We do not project functions based on homology cutoff at all, ever. So we use names given by curators and all these tricks and
Yeah, you have to make a judgment based on different types of evidence, blast being one of them. That's really the message.
So now we've all made a little subsystem, and the last operation for the day is the crucial one of saving it, so it's not any longer in a kind of a limbo and that it's actually archived. So I want you all to do this with your little subsystem. The way to do it is you should keep open the subsystem editor, so keep open that page and close everything else. I've been told that you really shouldn't have the subsystem and the different pages from the subsystem open when you save it. So close everything, but keep this subsystems overview page, and then click Manage My Subsystems. Now, this sometimes takes a while. And what you're doing is archiving your subsystem so it's stable. If you forget to do this after a day's work, it's probably not tragic. It won't automatically fall out of the subsystem or be swept away, you know, every 24 hours they clear it out or something. But you are vulnerable if the, I don't know, if the seed doesn't update between when you made the subsystem and when you've saved it, then you'll lose it. So what you should get when this screen comes alive is you should have a series of subsystems that you can manage, and you should only have one in yours actually, unless you're already a seed user, in which case you'll have more than that. Has everybody got a one subsystem page? Anybody not got anything? Okay, so by your subsystem, check it off in the square box. Scroll down to the bottom of the page, and there's these buttons that say Publish Subsystem to Clearinghouse. And that's the one that you push in order to archive it. And hey, this was quick. I mean, that can take half an hour sometimes. I'm impressed. So that's the crucial operation of the day. So nothing you've done should be lost. Now, I think that's where we're going to stop for today. If you've already got a subsystem of interest, you already know enough about the seed tonight to start playing a bit. I mean, you could start trying to build a subsystem if you know what you want to do, and that's great. You already know enough in principle to start.
One thing I want to add from doing this a lot, normally tonight, you're totally confused. You don't know where we're going. It's like, oh, it's too complicated. Why am I here? You know, why did I even think of coming to muggy Florida in June? But I guarantee that tomorrow, things will start to click. This happens, I would say, for 90% of you. But suddenly tomorrow, that's why it's so important to have you working on specific examples. When you start doing it on things you know, then you'll start getting it. I do guarantee that from doing this a lot.
Yeah, and another thing is that we've taught you some very basic operations today, and about 7 years ago I came to a workshop like this. I sat in that corner over there. I wasn't allowed to use the big computers, and I only learned as much as you've learned today, and then, you know, in my innocence and ignorance, and with Valerie at the end of a phone to help me when I had questions, I then took off. I mean, that was all it needed, actually. You can start quite modestly with this.
Yeah.
I mean, it's like, has lots of bells and whistles that are really powerful if you know how to use them, but you actually don't need to do that to begin to do significant work. So be confident that you don't have to master everything. The basics today is enough to get you started to doing something significant. Okay, thank you all.